Here we talked about the central dogma, the gene expression and the central dogma. And this is exemplified in this picture. The DNA, there is always a linear flow of information from the DNA to the RNA and from the RNA to the protein. And this is what is called as the central dogma. And during this process, the DNA, the information that is contained within the DNA, is first transferred to an RNA molecule, and that RNA molecule is later, the information is decoded by the ribosome and it is into the protein sequence. This is what is called as translation, and this linear flow of information from DNA to RNA and from RNA to protein is the central dogma, which holds true for all living organisms except for two of those viruses. The one is the HIV, and the other one is the hepatitis B virus, where this linear flow of information is not, it is not present. It doesn't occur in this way. Yes?
Those are the only two? Or
those are the only two members of the retrovirus family. They both belong to the retroviruses. HIV is the RNA virus with the RNA genome. Hepatitis B is the one with the DNA genome, but they both are the exception to this central dogma. The DNA inside the cell, some of the features of the DNA is inside the cell, it always exists as a linear polymer of the deoxynucleotide. I put a question mark over the deoxynucleotide because those deoxynucleotides are made up of three parts. There is a nitrogenous base. There is a nitrogenous base, which could be either the purine or the pyrimidine. The purines are made up of, the purines per se, the purines are made up of A and G. The purines are made up of A and G, and the pyrimidines are made up of C and T. That's how they are, usually they are present. Besides a nitrogenous base, there is a phosphate group. And along with the phosphate group, there is a pentose sugar, which is the deoxyribose. So together, 1, 2, 3, they make up, in a polymeric form, they make up the deoxynucleotides. And the two strands of the DNA helix, the two strands are in a helix-like structure, and they are anti-parallel. In the sense, if one strand goes this way, the other strand goes this way. It's like the divided 80 highway. One goes, traffic is flowing from the east to west, the other traffic is flowing from west to east. And this anti-parallel structure, it makes the DNA more stable. That's the best way we can explain the structure of the DNA based upon the space constraint that the two strands of the DNA are anti-parallel. If you look at one strand, it's over here, it goes from the 5' end. If you really try to trace it down here, that will be the 5' end, and the other end will be the 3' end. And the second strand will be, this is the 5' end. If you try to trace it all the way, you will notice that it will end up in the 3' end. And this 5' and the 3', those are the designated carbon of the pentose sugar. The pentose sugar has a 5' here, which is a phosphate group that has the phosphate group attached to the 5th carbon atom of this ribose of the pentose sugar. And any new nucleotide that is coming in it always comes and attaches to the free OH group at the 3' end. And that's why I say the DNA is always, it is getting extended in the 5' to 3' end always. Any new incoming nucleotide which comes in, like this shows a new nucleotide coming in, it will come and attach to this 3' hydroxyl, 3 hydroxyl group, and it will form a phosphodiester bond. So in a DNA, always the DNA synthesis occurs, it always occurs from the 5' to 3' end, from the 5' to 3' end, and these both the 5' and the 3', they are just a representative of the 5th carbon atom and the 3rd carbon atom of the deoxyribose or the pentose sugar. With this background, you also should be aware of the fact that the backbone of the helix is made up of the sugar. The backbone is made up of the pentose sugar, and the phosphate group. It's like a spiral staircase. You can envision the DNA as a spiral staircase, and the steps of the ladder, the 4 bases, that means A, C, G, and T. Which one? The DNA one? I posted it last week. Check it out, If not, just hear it and then you can transfer it. The 4 bases, that is A, C, G, and T, that form the steps of the ladder. So you can think about the DNA as a circular staircase where A, C, G, and T, they form the steps of the ladder. These are the railing or the backbone of the DNA sequence, of the DNA strand. The other thing to know about this is that the base pairing occurs in a very specific way, in the sense the purine base always pairs with the pyrimidine, or A will pair with T and G will pair with C. You will never have a purine base pairing with purine or the pyrimidine base pairing with pyrimidine. This never occurs in the DNA sequence. In all living organisms, we have never come across where purine is base pairing with purine or pyrimidine with pyrimidine. It's always a purine base pairing with pyrimidine in the DNA structure. And that's why the 2 strands are complementary. If you know the sequence of one strand as A, C, G, G, A, you can always find out what is the sequence of the other strand, because the 2 strands are complementary to each other. You get it, folks? The 2 strands are complementary. But here is an important part. The 2 strands of the DNA, they are joined to each other by hydrogen bonds. The 2 strands of the DNA, they are joined to each other by this hydrogen bonds. As is shown over here in this structure, these ones are the hydrogen bonds that connects, basically that is connecting this strand as well as the other strand. And my question is, if the 2 strands of the DNA are held together by hydrogen bonds, you tell me hydrogen bonds are what, strong bonds or the weak bonds? They are weak bonds. How do you explain the fact that DNA, which is such an important molecule, is the blueprint of life? You are what you are because of your DNA. You inherited that DNA, you will be passing on that DNA to the future generation. How come it just doesn't make sense that the DNA, which is such an important molecule, is the blueprint of life, but the two strands are connected to each other by such weak bonds. How do you explain that?
Is it split apart for forming new DNA?
They may form apart, or they do separate. They do separate when during DNA replication, during this transcription process, the two strands do separate momentarily. But you can also separate the two strands even if there's a covalent bond. How do you explain the fact that DNA, which is such an important structure, the two strands are connected to each other just by hydrogen bond? It's only, it's only hydrogen bond is basically connecting the two strands. Yes, William?
Probably what you're saying, but also there's like thousands of these bonds, so collectively you're gonna make it.
Good job. The answer to that lies in a more like layman terms. Just think, if I go and catch a simple penny or a cent from every person in continental US, am I going to become rich?
Yeah.
I will. Believe me, folks, if I just go and get about 10 cents or a penny from every individual, every soul in continental US, or if I say across the globe, then I'll become rich.
That's true. Mm-hmm.
Okay? And that's exactly because the DNA has so many of the hydrogen bonds together, it basically strengthens. It's a number. It's a number game. By all in all, you just take the example away and you just go into like a penny, you become rich. Now the DNA structure is susceptible to denaturation, and the denaturation is when the two strands separate. That's what is called as a denaturation. If you heat it at about 80, 85, or 70 degree also, the two strands of the DNA separates, and that's because the hydrogen bonds break. And that's what is called as a denaturation, or the melting of the DNA structure as such. An important aspect to know is it's not the number which is important, but it is the sequence of nucleotide that makes it much more valuable to the DNA. A DNA structure which is about 1,000 base pairs, but if those base pairs are in a random sequence, it's just a piece of trash. Or just a piece of DNA which is 100 nucleotide long, but those 100 nucleotides are arranged in a specific sequence. That is the one that carries a lot of meaning for the cell. So in short, the sequence of the DNA is much more important. Sequence of nucleotides carries more meaning than just the mere number of nucleotides in the DNA structure. DNA, every time the DNA is replicated, it is always distributed equally between the daughter cells. It never happened that one daughter cell receives more of the DNA compared to the other daughter cell. It is always universally distributed equally between the daughter cell when the cells divide. And just before the cell divide, there will be the replication of the DNA, which will be the next step. What I'm going to talk about is the replication process. And the DNA replication, there are several of those. There are 3 steps of the DNA replication, and a lot needs to be talked about the DNA replication. First of all, replication involves the breaking of the hydrogen bonds, which is technically called as unwinding of the DNA. And once the bonds break, there is a complementary base pairing. That means new bond, the new bases complementary to the template DNAs are being arranged, or they are being, they get into form the hydrogen bonds. And lastly, the bond formation takes place, and that bonding is 2 types of bonding. One is the hydrogen bonds between the bases, and also the phosphodiester bonds. So that bonding occurs, the phosphodiester bonding as well as the hydrogen bonds between the base pairs. Now there are specific properties of the DNA replication which is summarized here. Let me go one after the other. The replication is semi-conservative. It is not conservative replication, it is semi-conservative. That means two of the DNA strands, the two DNA strands which are made, they always have one strand as the original strand. Because this is the DNA strand, these are the new DNA structures which have been made to be transferred to the daughter DNA, to the daughter cell. Each of those DNA will have one of the original strand, and the other strand will be the newly synthesized one. One strand serves as a template for the new strand. So once the strand gets, the two strand gets separated, one strand will serve as a template, and the new strand will be made opposite to the template DNA strand. So it will occur both ways. The next one is DNA replication is asymmetric. What I mean by that is, during DNA replication, one strand is synthesized at a faster pace, which is called as a leading strand. And the other strand of the DNA is synthesized at a slower pace, which is called as a lagging strand. So it's an asymmetric process because there is a leading strand. There is a leading strand which is synthesized at a faster pace, synthesized at a faster pace, and there is a lagging strand. There is also a lagging strand that is synthesized at a slower pace. So this is what is the asymmetric DNA replication. The two strands of the DNA are not synthesized at the same rate. That's the take home. And because of that, you have the leading strand and you have the lagging strand. DNA replication is bidirectional. It starts at it starts at the origin, and the two strands, because this opens up, and the DNA replication begins at these two replication forks. It goes all the way. Eventually, it will get up to this point, and there will be the. Is everybody done there? Few more? Can you? Okay. Well, I'm also going to stop this 5 minutes. So this is what is the DNA replication is bidirectional. It occurs in two different directions at the same time. It starts not randomly from any place. It starts from a specific sequence. 1 second. 1 second, Jan. You have taken this one? I have to tell you something. There's a, just hold on for a second. Take a seat. So it starts from the origin and then it proceeds in both directions. Eventually, the two newly synthesized strands will separate. The last thing in this one is the DNA replication is a very accurate process. Mistakes are seldomly made during DNA replication. So it's a very accurate process, and that accuracy of the DNA replication process is due to the proofreading activity. Is due to the proofreading activity of the DNA polymerase. This is due to the proofreading activity of the DNA polymerase. You have this accuracy in the DNA replication. Okay? Folks, I will continue with the other one, but I want to make one thing clear that I will be here on Wednesday.